So welcome to the second Q&A session for operational tree improvement. My name is Blaise Ratcliffe. I'm a postdoctoral fellow at University of British Columbia, and I'm joined by our speakers here, Francois, Iman, Jean-Philippe, and Omnia, who all have excellent presentations. So I hope you've had a chance to check them out already. Have some questions prepared for them. We'll just open up the Q&A window here so you can submit through the official Q&A platform, or I'm also monitoring the feed loop chat. So if you'd like to use that to ask questions as well, you can. So let's start with a question for Francois. That's a question from Rita. Can remote sensing technology be used to evaluate tree health condition?
Hi. Yeah, thank you for that question. The answer to that is yes. The focus on the research that I was talking about at the beginning was using LiDAR, which is not necessarily that helpful for tree health, but some of the spectral data that we get from digital aerial photogrammetry is very, very useful for that. We can produce spectral indices to kind of help monitor them. The one example would be looking at tree pests, so spruce budworm in Ontario. That's a study that we've done earlier. I can link it in the chat, and that kind of outlines some of the uses for monitoring pests specifically.
Okay, and it looks like Antonio had a question that was similar to the first one, so I'm just going to check that off as answered. Let's see. Okay, and Antonio has another question for Francois. It's a multi-part question. Could you give some insight into the different genetic levels between individuals? So I think they're talking about the sort of top cross and versus wild. And are you talking about differences between individuals from genetic stands or populations, or individuals from the same site but with varying levels of kinship?
So to answer the first question there, we had 3 genetic gain levels. The first was the wild stand, which represents 0 gain. Then we had a mid gain, which represented 10% gain. It was 10% volume gain, and a top cross with 18% volume gain. And we were comparing the differences between individuals across those populations. We weren't looking at it in more depth. That's kind of the research that we're getting into now, to try and tie the remote sensing in with individual trees and using individual tree breeding values. So that was kind of our first introduction into trying to see how we can use remote sensing in tree breeding trials. I hope that answers your question. If you want more information about the differences between individuals, I can also, if you just send me a message, I can link the paper that outlines where these specific trees come from as well, and when they were planted. They were planted in 1996, I believe.
Thanks, Francois. Okay, I'm going to switch it up a little bit here. There's a question for Jean-Philippe from Beth. What did you find when you looked at different height selection scenarios in terms of affecting drought resistance?
Hello, Beth. Thanks for the question. Yeah, so the first scenario we looked at was 100% for selection for the height, and half of the drought response threats we studied. So the resistance and resilience were small, so were negative a bit, but the recovery was positive when selecting for only for height. So normally the tree with the high growth on height were less resistant but were recovered more after drought. But the integration after of wood density and drought resistance made it so we can all the threats were positive. So we had a genetic gain positive for the threats studied for all the threats at the site Normandin and for resistance at the site Watford, which was the only drought response threats we could study.
Thank you. We have a question from Marie for Omnia. Hi, Omnia. I was really interested in your pedigree reconstruction work from the polycross or polymix breeding. One question, did pedigree reconstruction result in any pedigree corrections?
Hi, Marie, thank you for your questions. And the answer is yes. So so the pedigree error, it ranged from 12% to 30% because we found unexpected pedigree errors. So we found that for 8 out of the 26 polycross family, we have 2 possible genotypes within this family. So there is 2 group of related individuals within this family. And this can be justified by 2 hypotheses. The first one, that the breeding was done on 2 clones. One of them was mislabeled. And the second hypothesis, that maybe the craft also produced cones. So now you have on the same tree 2 different genotypes. So when we are accounting for this unexpected error, the pedigree error now is around 30%. Yeah, I hope this is answering your questions.
Thanks, Omnia. The next question I'll take from Natalie. It's an open-ended question, and it kind of builds on a question that I also came up with. And so the use of quantitative genetics and genomic selection in combination with remote sensing, it can be a really potent formula to improve the efficiency of tree improvement programs. So what challenges or what difficulties do you see in incorporating these 2 technologies, the use of remotely sensed phenotypic data and genomics data? So that's open to any of the speakers.
I kind of have an answer. It might not be the answer that you're looking for, but it also ties in with Brian's question with the limitations. I think we need to take into account with this remotely sensed data is that it is just it's just a point cloud that you're getting. There's nothing to tie that aerial imagery with anything on the ground. And so one of the issues or challenges that we've had is to successfully link the data that we're getting from the remotely sensed data to what we have on the ground. And a lot of these trials were planted in the 80s, the 90s. We're working with one trial that was planted in the 70s, and they obviously didn't think, or maybe they didn't know that this was what we were going to do with these trials. And so some of the ways that the trials are measured, for example, it's not accurate enough, and we've had to go back there and take more GPS points, kind of try and figure out what things look like from the ground, because we can't just use, we need something to tie these 2 things together. And I think that's where the real challenge is. The younger the trial is, the easier it's going to be because we can automate more of the process. But I think that's kind of the real challenge, is making sure that we get the right tree from our imagery and connect it to the right tree on the ground.
Thanks, Francois.
If I can add something, with the more accessible cost of genotyping, more and more, like satellite imagery or drone imagery becomes like phenotypic value become the limitation. So when you can get like broad or easy data or on a phenotypic level, it becomes more and more interesting. But like Francois said, it needs to be accurate. But when it will be accurate enough, it must be really powerful.
Thanks, Jean-Philippe. I'm going to take a couple questions from the chat here. One question for Iman from Mikel. Mikel works in wood quality, but in radiata pine. And they would like to know, how did you measure wood density, and have you used a resistograph device before?
So the measurement we use for obtaining that density and ring width also was using X-ray densitometry.
Thanks, Iman. And another question that's been forwarded by Patrick Lenz from the previous chat. This question's from Raphael at UBC. Omnia, do you expect the production of foliar extractives to be different between adult trees? So the training population and the target population. And how could this affect your predictions?
So thank you for these good questions. So yes, it will affect our prediction. We know that genomic selection model is predicting the target environment at the same age and the same environment of your training population. So for our target population, it's measured in a different age, 1 year old seedling, and also it's measured in the greenhouse. While my model is predicting the breeding value at age 15 for foliar traits and in the site environment. So my expectation, this is why we have kind of decrease in prediction accuracy, because the level of relatedness is good enough to maintain the same prediction accuracy in the training population. But even with having different age, different environment, our prediction accuracy is relatively good. So I hope this is answering your question. Thank you.
Thanks, Omnia. Interesting question in the Q&A window from John Degner for Francois. Do you think LiDAR would be suitable for assessing canopy density as a proxy for Swiss needle cast, which is a foliar disease that causes needle shed? If so, what point density would you expect to be suitable?
That's a very interesting question. I think in theory, yes. At the moment, some of the UAV LiDAR systems that are being flown, even in our lab, we're getting over 1,000 points per meter squared. And so I do think that that's enough. You will have quite significant signal attenuation as you go down the crown. So we will have a lot more information about the upper crown. So as long as your area of interest, I would say, is high enough up the crown, that then I would think that it's valid. I do think it depends on how subtle the density differences are. So we found that even if you you have to fly your UAV in the same fashion for all of your sites and all of your flights, just to make sure that you have the same point profile through the crowns. And so I think as long as you can control all of those things, I would say yes. And probably I would think that UAV LiDAR that you can get from standard vendors would probably be high enough density.
Thanks, Francois. And another two-part question from Brian. They kind of build on each other. So in the assessment of progeny trials for assessing growth traits with UAV and LiDAR, are there any limitations based on tree age and planting density? And the second part to this question is, and you kind of already touched on it a little bit, Francois, what needs to be taken into consideration when planning new experimental trials now that we have this technology, this remote sense technology readily available?
So for the first one, yeah, I kind of touched on that before. That is the issue that we've run into with some of these older trials, is that there's just a general GPS point from like the mid 90s saying, okay, this is row 5, you know, and it gives you a general location. I would say we need to be very clear about where these trees are on the ground. And so that might mean putting ground control points down when we collect the data, and just really being able to, so that we can reconstruct the grid to match with our trees. So that's why I was saying that the younger plantations are much easier when crown closure hasn't occurred. We want to try and automate as much of this as possible, which means that we want to have good tree top detection, which as you increase the stand density, it becomes more difficult. As the stand gets older, you also have other things that are growing in the stand. You have places that were unplantable positions that now might have a different tree species growing. And so these are all things that we have to take into account as our trials get older. So we found our trial that was planted in the 90s, it's quite a bit easier to kind of separate out the trees that we knew that should be there versus the trial from the 70s. We're not entirely sure where all the trees necessarily should be. The other thing is to take slope into account. I know that we can't always choose where we plant these trials, but part of the way to really get accurate tree heights and stuff like that is to normalize the point clouds to ground level. And so if we have a steep slope and we're trying to plant trees on the slope, I think it's not that easy. If you're on the ground trying to plant things at 3-meter spacings or whatever, it's just tough. So I think taking that into account as well, those would be, I would just say that's the GPS is like the biggest thing, and the biggest helper for us is going back in afterwards and getting really accurate corners, identifying where the gaps are in the canopy so that we can reconstruct that later.
Thanks, Francois. And we have just a few minutes left here. I have one of my own questions for Jean-Philippe. So one of the interesting results of your studies was the strong G/E interaction for your drought resistance phenotype. And so what methods would you consider using in the future for future studies to explore this G/E interaction for drought response traits? So if you were planning a study now, what methods might you use to elucidate those interactions?
Yeah, so there are several factors that were different at both sites and both drought events. So what I'm thinking about is the age of drought, but certainly the timing of the drought and the growth period. So there needs to be several studies that take that study like individually, each step, each factor, so we can like split all the variance components when we need to analyze the drought events on several sites. So yeah, we need to analyze several drought events at several timing in the growth period, so we can like correctly assess the genetic by environment component. So we on our side we shot that we proposed this interaction, but we did not analyze it properly.
Okay, thanks for the very good answers. So I think we're right on time here. Let me check. Yes, right on time, 10:45. So with that, I'd like to thank all our speakers and also everybody who asked great questions. And I look forward to seeing everybody in the next session. Thanks, everyone.